my talk will be about very special cell type in animal body. My talk is titled Inner Amoeba. You will see why it's amoeba. So first, I would like to introduce you to the distinction between soma and germline in animals. So imagine that this is a fertilized egg, a zygote, and it goes several cell divisions, so 2 cells, 4 cells, 8 cells, and so on. And the embryo is developing, and at some point, the lineage specifications starts working. You have like cell progenitors, which in turn give you different cell types, for instance, muscles or neurons, which are all collectively called soma. But very early, there's a branching of this pathway of differentiation where some cells, actually very early before this happens, are separated in the embryo. They are called primordial germ cells, and they look like small amoebas, basically. And these cells will eventually give rise to germline, so to sperm and oocyte. And this lineage is called germline. And of course, it goes on for the next generation and next generation. So overall, if there are any mutations happening in the soma, they are never passed to the next generation, whereas mutation in the germline will affect the offspring. So I don't have time to talk about it, but it's like amazingly interesting immune system in this germline. They are very, very special, these cells. And these primordial germ cells, we know how they are formed. So we work in zebrafish, and in zebrafish, there is something called germplasm, which is basically a gooey substance with lots of proteins and lots of RNAs, and it's deposited already into the oocyte. And where the embryo divides, so here we have 4-cell stage, you see these bright dots here. So these are markers for the germplasm. So at the 8-cell stage, you still see 4 of these dots. So basically, not all the cells get the germplasm, so not all of them will become a germline. The rest will become soma, of course. And this is, all these experiments are done by Fabio D'Orazio, my colleague. I am a computational biologist, so it's more about my computational work with this data. So after many more cell cycles, these germ cells, these primordial germ cells, they locate into like 4 corners of the embryo, and around 3 hours post-fertilization, this germplasm starts getting fragmented, and then around 4 hours after fertilization, so into gastrulation already, these cells start to migrate. So from these 4 corners of the embryo, they, by mechanism of chemotaxis, they navigate towards genital ridges in the embryo, in the developing embryo, where they will form germ cells, so to the future gonads, basically. So at this point, their task is basically to migrate from one place to another place. And since we are in the era of deep sequencing of everything, so this project is about characterizing the differences in the genome-wide scale of the gene expression and the chromatin between somatic cells at earlier and later stages, and between these somatic and PGC cells, and we do it in zebrafish. We do it in zebrafish because, of course, in human, from ethical reasons, working with embryos is not possible, and in mouse, due to technical reasons, it's also hard. In contrast, in zebrafish, The eggs lie in water. They are transparent. You can put them anytime you like in the microscope. You can do transgenics. All kinds of things are really working very well in zebrafish. And it's still a vertebrate, so it's not too far from human. OK, so I need to tell you a little bit, just one slide about the regulation of gene transcription in animals. There are many, many different processes that contribute to it, like DNA methylation, histone modification, and so on. But the most important part is the assembly of this blob, which is an RNA polymerase II complex, which covers around 100 nucleotides on the genome on a place called promoter. So the gene will be on the right side here, and it's regulated by these proteins. They're called transcription factors. They bind to the DNA, which can be nearby the promoter, or they can be far away, like the DNA can form loop. It can go up to millions of bases away, and then it's called enhancers. So enhancers are present in multicellular organisms. In unicellular, these almost don't exist. And I will tell you about the open regions in the both cell types. So to pinpoint these regions here, where something is bound, the easiest way to look is to look where the chromatin is opened. So when the protein binds, it cannot be a nucleosome there. Nucleosomes cover the DNA. So there is this pretty new protocol, which is called ATAC-seq, which basically chops the genome at the places where DNA is opened and accessible to other proteins by using a transposase. So it's one of these biological hacks when you use some machinery and you repurpose it to use it as a tool for the experiments. Like CRISPR is one of the top ones right now. And this is another one, also very bright way of using existing proteins. So when you do this kind of experiment and you sequence it afterwards, what you look at is this kind of a plot, lots of genomic browser plots where you see the signals. So in this case, we have a gene here, and at the promoter there is a big spike, which basically means promoter is open. That's what you actually expect. And in some cases, you have more complicated view, like with a lot of other peaks, like maybe this one is actually an enhancer, we don't know. So at 3 hours, so at 3 hours, I compared the levels at the somatic cells with with the levels at PGC cells to produce a plot. And what I got, basically a diagonal, correlation coefficient 98%, meaning that basically there is no differently opened regions in PGC and Soma. So kind of a depressing view because we didn't find anything. And then I looked at maybe there is a shape difference instead of just levels. And actually, if you look at different classes of genes, they somatic in red and blue in PGCs, they look very, very similar. So also no shape difference. But then, when I looked at 24 hours, some miracle happened. I got this plot. So basically, 2 diagonals here, which when you compute the difference between openness in these regions, you get this kind of a histogram. Okay, so I was expecting one Gaussian with at 0 and maybe some tails, and then you run your statistical test, you get the tails of the distribution, there will be something. Instead, I got 2 Gaussians, which is like amazing, absolutely amazing. I never seen such thing before in computational biology. So I maybe thought it's some artifact, maybe some chromosomes are heterochromatinized and maybe not accessible, maybe there's some global feature. But if I analyze the genome globally, I don't get this shape. I get one single Gaussian at 0. So we did lots of replicates. So whenever I'm comparing 2 somatic cells, 2 somatic samples or 2 PGC samples, I'm always getting one diagonal. and when I'm comparing across, I'm getting 2. So I think that the thing is real. So next thing I did was a principal component analysis of this chromatin opening. So it's a way basically to visualize your samples in like 2-dimensional, in this case, plot. So we have somatic and PGCs late and early. So all early cells, they cluster together. We know that there is no difference between 3 hours, this and that. So exactly it's shown on this plot. And when the time passes to 24 hours, PGCs and somatic cells, they are separated, but not much. They come close to each other. And that is when I look at the promoters. But if I look at the distal elements, enhancers, something completely differently emerges. So the somatic early and PGC early, of course, they cluster, but then they go almost orthogonally to each other. So PGCs and somatic cells are completely separated in this case. So there is something going on that is real, and then it's a very strong effect. I also looked at the regulatory these regions surrounding known PGC and known somatic genes. So basically, the more down it is, it's more PGC-like, and at the top it is there's more density of the somatic genes. So if you are far away from the gene start, it's basically around 0, this profile. And for Soma, it stays 0 for somatic genes. But for PGC genes, there is a big depletion in these somatic elements, meaning that actually in the range of tens, maybe to hundreds of thousands of nucleotides, there is a depletion of these regulatory somatic elements around them. And then I asked the question, so what contributes to them? What opens them? What kind of proteins are bound to these regions? Unfortunately, we work with zebrafish, where the data is very sparse compared to human and mouse. So I had to do predictions. So I proposed a very simple model where the openness of a genomic region I discovered can be displayed as can be modeled as a sum of more than 300 transcription factor binding motifs, so the DNA sequences, times the function which is 1 if there is a site for a given motif, or 0 if there isn't. So by this way, I selected which motifs will be responsible for these PGC-like elements in one diagonal versus the one on the opposite diagonal, which is the somatic ones. So the somatic ones, there are loads of interesting transcription factors. They all make sense. Zinc fingers, nuclear receptors, and so on. They are all important for the developing embryo for different tissues. But for these PGC-like elements, I've got only 4 motifs. So, and 2 of them, E2F and NRF, they are very general motifs, and the other 2 are so CTCF and Boris. They are structural proteins. So CTCF is known to known to bind hardly to the genome and provides like scaffolding points for the chromatin structure. So not really regulatory. It doesn't induce gene expression on its own. And overall, using this model, I managed to explain almost 40% of the variance in the data, which is amazing, because I was doing these kind of things before for transcription factors and promoters, and I never got anywhere more than 15%. If I got 15, it was amazing. I got almost 40 here. So it's really like I'm capturing almost everything using these motifs, and the CTCF is on top of it. And actually, for the CTCF, there is the data. Exists. It's a poor quality, but it's there. And so I can plot the true biological binding of CTCF and its partner, RAD1, at this region. So you can see that there is much more binding of CTCF at the PGC-like elements than in the somatic elements. Okay, so that was my last slide. So in summary, gene promoters in PGCs behave very similar way to the somatic cells, regardless which stage you look at. But there is a gigantic difference when you look at distal elements. So regulatory elements which are bound by transcription factors are way, way down-regulated in the PGCs, but not the structural elements which are bound by CTCF and its partners, which are open in both cell types. And the vicinity of the PGC genes is also depleted for these regulatory regions, which makes totally sense in this case. So the outcome is that PGCs not only crawl like amoeba in the developing body, but they also behave, the regulatory mechanisms are like in the unicellular organism as well. So if you look at this picture that I showed you in the beginning, where we have these transcription factors here and the looping, it's essentially what happens in PGCs, that this distal looping probably doesn't exist at all. So the chromatin structure is something completely different from the somatic cells. Okay, thank you very much.